everybody, it's Miss Hamill. I'm here today to talk to you about energy. Really, we're going to focus on enzymes for our last chapter or our last lecture on the chemistry unit. Okay, so metabolism is going to be the totality of an organism's chemical reaction. So what goes into that organism and what comes out. And it's the management of materials that go in and, again, the energy resources of this cell. Catabolic pathways are going to release energy by breaking down complex molecules into simpler ones. So that would be like taking a long starch molecule, like the ones we built in class, and breaking them down using hydrolysis. Digestive enzymes breaking down food to release energy is an example of a catabolic pathway. So a key thing is catabolic pathways release energy. Anabolic pathways are going to consume energy to build complex molecules from simpler ones. So that would be taking a glucose molecule using dehydration synthesis and putting them together to form a big starch, or amino acids linking together to build muscle protein. Energy is going to be the capacity to do work. And there's different types of energy. The first one is going to be kinetic energy, and this is any energy associated with movement. So when we run, when we jump, even the movement of particles throughout our cells. Heat is thermal energy, and this is a specific type of kinetic energy, and it's associated with the random movement of atoms or molecules. So as you may remember from middle school, or even before that, when it is cold, the molecules are going to move much slower, and when it is hot, the molecules are going to speed up, creating the heat that we feel. Potential energy is going to be energy stored as a result of its position or structure, and chemical energy is going to be the energy, potential energy that is available to release in a chemical reaction. So energy can be converted from one form to another. For example, it can be taken from chemical energy, so the food that we eat is broken down so that we have the energy to do physical work, or mechanical energy, and then electrical energy, so the signaling of our muscles. Okay, here's just an example. The diver on top of the diving board is going to have more potential energy than the diver below, or the same person. This is a little circle. So when the diver dives into or jackhammers into the water, it converts that potential energy into kinetic energy or motion. And then again here, when it's climbing up the when he's climbing up the ladder, he's going to convert the kinetic energy back into potential energy. Thermodynamics is going to be the study of how energy flows from one form to the next and the transformations in nature. And there are some laws of thermodynamics that you need to know. So before we get into those laws, a closed system is, think of a thermos. So if you have a thermos and you fill it with hot coffee, the liquid in the thermos is going to stay hot, and it's isolated from its surroundings. It's isolated from the air around, so it doesn't cool down. An open system, energy can flow from one system to the next, so one system to its outside surroundings. So organisms are open systems. We are going to have a flow of energy. Think of the food webs that we talked about earlier, and how energy flows from one organism to the next. So the first law of thermodynamics is that the energy in the universe is constant. So energy is going to be transferred and transformed. However, it is never created or destroyed. So this is called the principle of the conservation of energy. So energy is going to be conserved. So this bear here, he is going to eat the fish, which is chemical energy, and then it will turn it into kinetic energy to move and function, carry out life. Okay, the second law of thermodynamics is that every energy transformation increases entropy or disorder of the universe. So during every transfer or transformation, some energy is unusable and is lost as heat. So if I want you to think about the food pyramid again, or the energy pyramid again, when we go up the trophic levels, we are only getting about 10% passed on from level to level because it is often lost as heat and through metabolism. Okay, free energy is going to be the part of a system's energy that is available to perform work. And delta G, or change in G, is the change in free energy available. So exergonic reactions, energy is going to be released. This is a spontaneous reaction, and delta G is going to be less than 0. Endergonic reactions are energy is required. They are going to absorb free energy. So you can think of the sun and plants absorbing that energy. That's an endergonic reaction. And delta G is greater than 0. So if you take a look at these graphs, you can see here that in exergonic reaction, the reactants are greater than the products because energy is being released. So the amount of energy released here, delta G, is less than 0. And then here you can see that in endergonic reactions, the reactants are less than the products, and there is an energy input, so it's absorbing energy, and the amount of energy required, delta G, is greater than 0. So a cell is going to do three types of work: mechanical, transport, and chemical. And a cell's going to do what's called energy coupling. So it's going to use exergonic processes to drive an endergonic process. So a cell is going to use the ATP from cellular respiration to drive other functions in the cell. ATP is adenosine triphosphate. This is going to be the main energy source in the cells, or an energy coupling. ATP is adenine ribose, which you should remember from RNA, and 3 phosphates attached, so 3 phosphate groups. So this is the structure here. So when bonds are going to break between the phosphates group through hydrolysis, energy is going to be released. And the release of the energy comes from the chemical change, so the state of the lower free energy, and not from the actual phosphate bonds themselves. So here you can see that through hydrolysis, we have 3 phosphates, or ATP, and then it's broken down into ADP diphosphate, so there's only 2 phosphates left, and now we have energy. So from the breaking of the bond of the ATP, we get energy and ADP. So how ATP performs work: that exergonic reaction releases the PI, which is used to do the exergonic work of the cell. So it releases the energy in the phosphate. Okay, so when ATP is hydrolyzed, it becomes ADP, so adenine diphosphate, which I just said, and this cycling here is going to drive other chemical reactions in the cell. So it'll take the energy created when ADP and energy is formed to drive the cellular work. Okay, here are some examples. So motor proteins, they're going to use some ATP to do the muscle movements and work. So here we have a transport ATP. They're going to transport proteins into and out of the cell, protein and other substances, using ATP. And then here we have just binding of different molecules, and it requires ATP. Okay, so catalysts are going to be a substance that change the rate without being altered in the process. So they do this by lowering the activation energy, or the energy required to start the chemical reaction. So they speed up these reactions by lowering the activation energy. So enzymes are biological catalysts, and they most often end in ASE. So if you see a word that ends in ASE or ACE, it is most likely going to be an enzyme. So sucrose, which is a sugar, is going to be broken down by sucrase. So you can see here, sucrase is going to speed up the chemical reaction and going to form glucose and fructose. Now this is going to happen regardless of the presence of sucrase. However, with the addition of sucrase, it's going to speed up this chemical reaction, make it much faster. So this is a hydrolysis reaction because you can see we're adding water and we're splitting apart the molecule. Okay, here you can see that we have the chemical reactions. The catalyzed or the reaction with the enzyme is in red, and the uncatalyzed reaction is in black. So you can see that the amount of energy required to start the reaction is going to be lower in the one with the enzyme. The amount of products at the end is going to be the same. Okay, so enzymes are very specific, so they're going to act on one part or one substrate. So it's very specific, so only one enzyme for one type of substrate. The enzyme is going to bind to the substrate, and this is the reactant of the chemical reaction, and this is called the enzyme substrate complex. The active site is going to be the region where it binds, so I like to think of Pac-Man. So it's going to be the mouth of Pac-Man. And here's a little example here. You can see that here's the active site here, and if we had an enzyme substrate, it would bind right into that little piece. So here's an example. Here's the enzyme substrate complex. You can see the substrates are here. They're very specific. They have a specific shape that matches up with the enzyme. They are going to bind with the enzyme and the active site, which is right here. And then it's going to speed up the chemical reaction by lowering the activation energy, or EA. And the substrates are going to be converted to the products. The products are released, and then this enzyme is available to bind with new substrates. So induced fit means that the enzyme is going to fit snugly over the substrate, and it's like a handshake or a lock and key you can think of. Very specific. So enzyme activity can be affected by temperature, pH, and chemicals. So like we did in our lab, you saw that the temperature, increase or decrease of temperature, or increase or decrease of pH is going to alter the enzyme by denaturing it. So it's going to change the shape of the protein. Remember, enzymes are proteins. And then the substrate is not going to fit into the enzyme as well. So the chemical reaction won't occur as quickly. So there are cofactors, and these are helpers to enzymes, and they could be minerals such as zinc, iron, and copper. Coenzymes are going to be organic cofactors such as vitamins, so they are helpers. And then we have what's called an enzyme inhibitor. And inhibitors are going to either bind to the active site or another part of the enzyme to change its shape, therefore the chemical reaction would not occur. So competitive inhibitors are going to actually bind to the active site and compete with the substrate, and non-competitive inhibitors are going to bind with another part of the enzyme, changing the shape of the enzyme and the active site, making it non-functional. So this is denaturing of the protein sometimes. Okay, so this is normal binding. You can see that the enzyme and the substrate fit together. Here, a competitive inhibition. We have a competitor that fits in, blocking the substrate, and then the non-competitive inhibitor is going to bind to another part, changing the shape of the enzyme, making the substrate not fit properly. So to regulate these metabolic pathways, they can switch on and off the genes that encode for specific enzymes. We'll talk more about that in the future. We have allotristic regulation, which is proteins' function at one site is affected by the binding of another regulatory molecule to separate site or at a separate site. This is the allotristic site. And the activator stabilizes the active site, and the inhibitor stabilizes the inactive form. And cooperativity was when they changed